So we're moving on to chapter 4. So we've already talked about chapter 4 a little bit in lab. So we talked about the organelles that were in the cell, so make sure to review that. And we also talked about mitosis and cell division, or we will be talking about that. Hopefully you looked at that last week. So we've started with chapter 4, and now we're moving into the lecture portion, and we're going to talk about plasma membrane transport, or transport across the plasma membrane. So remember, the plasma membrane is going to be that outer covering of the cell, the outer layer of the cell, and it serves as a physical barrier between the cell and the fluid that surrounds it. We call the fluid that surrounds the cell, we can either call it the interstitial fluid, it means it's in between the cells, like in your tissues, or we can also call it the extracellular fluid, so meaning that it's outside the cell. And the fluid inside the cell, we can call that the cytosol. Remember, that's the stuff that's floating around, or we can call it the intracellular fluid. So it's the fluid that is inside the cell. And so this plasma membrane is going to be really important. It does a couple of things. It regulates the movement of substances into and out of the cell, right? So things that the cell needs to obtain, it's going to try to bring them in, and things that it doesn't want, like waste products, it's going to try to get rid of them. It establishes and maintains an electrochemical gradient, and we're going to talk about this in the upcoming chapters a little bit more, but it will establish these concentration gradients where the things that it wants inside the cell, it's going to keep a bunch of that inside the cell. So we'll have a lot inside the cell, but not very much on the outside of the cell. We call that a concentration gradient. Or, vice versa, if it doesn't want it inside the cell, like it wants to keep sodium outside the cell, we'll have a lot of sodium outside the cell and not very much sodium inside the cell. And that's going to be called a concentration gradient. Now, the electrical type of gradient is basically that the inside of the cell is usually a little bit negatively charged when we compare it to the outside of the cell. So it has both chemical concentration gradients and electrical concentration gradients across that plasma membrane. And remember, back to chapter 3, we said that the plasma membrane, because of these concentration gradients and electrical gradients, has some potential energy because those ions kind of really want to get back into the cell or out of the cell. They want to move down their concentration gradients, and we'll be talking about that later. The plasma membrane is also important in cell-to-cell communication, and we're not going to talk too much about that in this class, but or this lecture, but we'll talk about it more in our tissues lecture, and then as well as the endocrine system. So most of our cells are going to be, if our cells are in like the epithelial tissue, they're actually connected to each other. But cells can also communicate to each other with hormones or different cellular signals, and they're going to bind to receptors on the cell membrane. So membrane transport is what we'll be talking about for this lecture, and it's a really important process, right? We need to obtain things that the cell needs and eliminate waste products. And we have two categories of it, the passive processes and the active processes. Now, before we move on to that, I just want to make sure that you guys remember what the plasma membrane looks like, and that it is a phospholipid bilayer. That's the main thing here. So we've got these purple polar heads and these yellow nonpolar tails. And so the main portion of the plasma membrane is going to be kind of full of lipids, all these fatty acids on the inside here. And we do have these blue proteins, some of them that are embedded all the way through, those are integral proteins. Some of them that float on top or just inside, those are peripheral proteins. Anytime you see a carbohydrate attached to a protein, that together is called a glycoprotein. The carbohydrate attached to a lipid is a glycolipid. So just remember, go back to your our lecture about the cellular structures and go back to your lab manual and make sure that you can look and label this. So we'll talk a little bit about the plasma membrane, what it's made of. So it's definitely important to know that it's made of a phospholipid bilayer and that the inside of it is full of a majority of lipids. And so some things can slip between these and some things can't. All right, so this is going to be what we're talking about in this lecture. You can see it's kind of a lot of information. We're going to start over here with passive processes. Passive processes do not require the cell to expend energy. They're going to happen on their own. Substances are moving down their concentration gradient. They include diffusion and osmosis. There's a couple of different types of diffusion. Simple diffusion, which does not require a transport protein, and facilitated diffusion, which does. And we have channel and carrier mediated. We're going to spend probably the majority of the time kind of discussing diffusion and osmosis, and then we will also talk about active processes later in the lecture. And so active processes do require the cell to expend energy, usually in the form of ATP, but sometimes not. And these active processes are going to usually either move a substance up its concentration gradient, or it's going to actually form a transport vesicle. And these transport vesicles, that's this part over here, they're generally going to be moving really big things across the plasma membrane, or a lot of things that we wouldn't have been able to move all of those very easily otherwise. And it takes a lot of energy to move these vesicles around and form them. So all the vesicular transport is active. So we're going to go back and we'll talk about passive processes first. So passive processes do not require energy. So remember, ATP is the energy currency of the cell. We don't need that here for these passive processes. In these passive processes, substances are going to be moving down a concentration gradient. And so whenever we have a concentration gradient, remember I said earlier, it means that there is a difference in concentration of a substance in two different areas. So for example, I really like to talk about, like, let's say we're all sitting in a classroom, which we're not now, but we would be if this was an in-class. But we could all be sitting in a classroom, and if we're sitting in a classroom and we had, say, 80 people in this classroom, and then outside we have a door, right? It's a normal classroom, it has 4 walls and a door. Outside that door is an empty lobby, all right? Because everyone's in class now, no one's really out in the lobby, it's class time. And so all of these 80 people, it's like really hot outside today, it's like 80 degrees, the air conditioning's not working very well, and it's really hot inside. Because we have so many people in the one room and we have no one outside, if I open the door, do you think everyone's going to, like, once lecture's over, I guess, or if I open the door, is everyone going to want to just hang out in the classroom with 80 other people in this really hot classroom, or are they going to want to spread out and then go out to the nice lobby where there's no other people? Well, usually they'll want to spread out. So think about that as moving down a concentration gradient. So we had a lot of people concentrated in the classroom, and we didn't have very many people concentrated in the lobby, so people wanted to move from the classroom to the lobby, from an area of higher concentration to an area of lower concentration. And so this is going to be kind of the main driving force between these two different types of passive processes, diffusion and osmosis. And so we're moving from an area of more substance to an area of less substance. So everyone wants to kind of spread out and have their own personal space. And they're going to, eventually move around so that everyone has, it's about the same amount of personal space between them. So as we said, the opposite of passive processes are active processes. These require energy. And usually the substance is moved up its concentration gradient, or we're making a vesicle and moving it around. Both of those require a lot more energy because they don't really happen naturally. So the first passive process we'll talk about is diffusion. And once again, diffusion is the net movement of a substance from an area of greater concentration to an area of lesser concentration. So in general, whenever we have ions and molecules, they're in constant motion. They have constant, they move just a little bit. If you heat them up, they move a little bit faster all the time, even if they look like they're still. And if unopposed, all of these things, this just random movement of molecules, they're eventually just going to diffuse around, and they're going to eventually reach what we call equilibrium. And that means that we have all of those molecules are evenly distributed throughout a given area. They don't really clump together necessarily. They're all going to kind of spread out until they all have their own little bubble of personal space. They'll all keep moving around, but no one gets too close to each other, right? It's like we're all physically distancing. They're all staying 6 feet. They're all staying 6 feet apart if they can. They don't want to be too close to anyone else. Good, disease prevention tactics. So how fast this happens, and before I mention this, I want to say a good example of diffusion, and I did this with my daughters the other day, is if you take a glass of water and you put a drop of food coloring in it, and there's a picture in your book of this, and it might be coming up in another slide, I can't remember if I put it in, but the drop of food coloring will start together, and you'll have all the food coloring in one spot, and then, the clear water around, and eventually, if you just leave the glass alone, eventually the food coloring will spread throughout. Like if you leave it overnight, it's going to take a while, right? Because this diffusion is not the fastest, especially if it's cold. But what could we do to speed this up? Well, one of the things we could do is heat it up. So if we heat up our glass of water, or we put it in a glass of warm water instead of a glass of cold water, then that's going to make all of those molecules move faster. And so if they move faster, they can spread out more quickly. All right, so we have higher movement with a higher temperature and a faster rate of diffusion. The other thing is that there's a steepness of the concentration gradient. And so if the concentration gradient is very steep, meaning we have, let's say 100 molecules of salt in one area and 2 molecules of salt in the other, it's going to be like a really, it's a big difference. And so that makes a really steep sort of hill for this concentration gradient. And when that happens, our steeper gradient causes a faster ratio of diffusion. Let me see if I can pull up the whiteboard. That way I can draw for you. All right, maybe. No, it is not working. All right, well, maybe we'll be able to pull up the whiteboard in a later video. But I usually like to think of the concentration gradient as a hill. So you can see here as I'm moving my cursor, like this would be a hill. And if we have a really steep concentration gradient, say we had 100 of something up here and we had 2 of something down there, it's going to give us a steep hill, versus if we had 10 of something and like 5 of something, we'd have a much more shallow hill. And if we have a bowling ball at the top of that hill, which hill is the bowling ball going to roll down faster? The one where we have 100 in one area and 2 in the other, right? The steeper hill. The bowling ball rolls down the steeper hill faster, and that's one of those things that the steepness of the concentration gradient is going to affect how fast diffusion happens. So the bowling ball is going to roll down that hill a lot faster than it will roll down, the much shallower hill. If we have 10 molecules of salt in one area and 5 in the other, it's much shallower, and so the bowling ball is not going to roll down that hill as quickly. And so the other thing I like to mention is this is passive transport, so we're moving things from an area of high concentration to low concentration. If we put that ball, the bowling ball, at the top of that hill, will it roll down or will we have to push it? Right? I mean, as long as you're putting it on the very edge, it's going to roll down on its own. You don't need to push it down. Gravity will pull it down. It's going to roll down on its own. It's a passive process. We don't need to add energy. So here is the example I told you about putting the food coloring in there, and it's going to start out together, and then with that random motion, even if you don't stir it, it will eventually diffuse, right? So it'll eventually diffuse through. If you put more food coloring here, right, then we'll have a steeper concentration gradient. It'll probably diffuse a little bit faster, or if we heated it up, it would diffuse faster. So there's a couple different types of diffusion, and diffusion is going to be, we're thinking of like moving. Generally, we think of water as what everything's dissolved into, right, our solvent. And so when we talk about diffusion, we're thinking about things that are in water, that are dissolved in water, the solutes that are in water, so the salts or the sugars, things like that. And so diffusion is talking about the movements of those sugars and salts, like throughout the water. So simple diffusion is when we have molecules that pass between the phospholipid molecules. This is anything that does not require a channel. So simple diffusion across a cell membrane would mean that things can just cross the cell membrane by itself. I like to think of the cell membrane as a wall, right? So once again, if you think that we were in a classroom, think that we have the wall. The front of the classroom is where the door is, and our door will open and close, but generally, let's say that I closed it for lecture. So normally it stays closed. And then we have the wall. So the wall is our plasma membrane, and the door is like a channel or a carrier protein that can open or close to let things in or out. These molecules that can go molecules that can use simple diffusion are really usually small and nonpolar, right? Because they're nonpolar, they're usually lipophilic. They like lipids, so they can kind of pass through the lipids of that phospholipid bilayer pretty easily. And because of this, they are they're like ghosts. They could go right through the wall. They don't need to use the door. They can just diffuse right through the wall, diffuse right through the plasma membrane. They don't need a door. If you were trying to, put a ghost in jail, in a normal jail cell, they would just be able to float right out. And that's the same thing with these simple diffusion, with these nonpolar solutes. The cell cannot do anything about them. They can leave the cell or enter the cell just based on their concentration gradients, but the cell can't keep them in and it can't push them out, right? So if you had, a ghost haunting your house and you close the door, it could just fly right through the door. So that's what these little small and nonpolar solutes that can use simple diffusion. So oxygen and carbon dioxide, their respiratory gases, are a really good example of this, and we'll talk about this in 169 when we talk about respiration, how they're going to just move down or up, well, not up, they're going to move down their concentration gradient regardless, and the cells really can't, they can't do very much to keep them in, you know. If they're in the cell and they can just pass right through the cell, if the concentration gradient exists, if they can go down a concentration gradient, they'll either go into or out of the cell. So they can pass right through it. And so they're not regulated by the plasma membrane. And the movement is only dependent on the concentration gradient. And the movement continues as long as a gradient exists. But remember, if we started at, 100 things in one area and 2 things in the other, and things started moving from one side to the other, eventually they would reach equilibrium, right? At 51 things on either side, right? So eventually they'll reach equilibrium if they keep moving through. So small nonpolar solutes like oxygen and carbon dioxide, they go right through. They don't need a door. They don't need a plasma protein in order to move them through. They can just, they're like ghosts. They just go right through. So next we have, everyone else. Everyone who's alive and has mass and takes up space. We require something else. Like, we can't just go through the doors, go through the walls. We require a door. So most of our solutes are going to require a channel or a carrier, like a protein carrier, in order to get them through this plasma membrane. So for most solutes that are not so small and nonpolar, usually they're larger. They might be a big molecule like glucose, or they might have a charge. They might be ions. So they can't just, if they try to go through the wall, they would bounce back, right? And so they can't just go through the walls. They can't go through the plasma membrane on their own. And so they need a special assistance from plasma membrane proteins, right? And that's what I said, that door was like a plasma membrane protein. It can open and close to allow molecules to pass into or out of the cell. And we have two main types, channel-mediated diffusion and carrier-mediated diffusion. Channel-mediated diffusion is what we'll talk about most in this class, and this is when we have a protein transport channel. So this guy right here is a good example of a protein transport channel. You see how it has a hole in it? This hole here, when it opens, is going to allow certain ions or certain molecules to pass into or out of the cell. Once again, from an area of high concentration to an area of lower concentration. If we are a passive process, diffusion, everything is moving down its concentration gradient. And so these special channel proteins are going to allow, when they open, they allow the movement of small ions through these water-filled protein channels that I just showed you. These channels are specific for one type of ion. So you might have a sodium channel that only lets sodium in, or a potassium channel that only lets potassium in or out, or a chloride channel that only allows chloride to move through. And the channels can't dictate the direction that the ion moves. It can't say, oh, you can only come in or you can only go out. The channels are going to allow this ion to move down its concentration gradient. We have a couple different types of channels depending on whether they're open or closed or can be open or closed. The first is a leak channel. And I think of this as like one of those, like an archway or a doorway that doesn't actually have a door. It's continuously open, and it's kind of like just funneling people through that area. Leak channels are always open and things can leak through them. A gated channel is kind of like a door, or if you think about a farm, like a gate in, a fence, where usually they're closed. And they will open in response to a stimuli just for a fraction of a second, and then they close it back again. So I like to think of gates on a farm, because if anyone's ever been on a farm, you know that if a gate, if you opened a gate, you close it again. Don't leave it open, because if you leave it open, maybe the horses are going to escape and run out onto the road or something. It's never happened before. So you always keep those gates closed. And so the gated channels are usually closed, and they'll only open for a second in response to a stimuli to let a couple things in or out and then close back again. And these are really important in the normal function of muscle and nerve cells, which we'll be learning about in the 168. So we will talk about them again. Wrong way. So let's see. Channel-mediated diffusion, and now carrier-mediated diffusion. So carrier-mediated diffusion is going to be very similar to channel-mediated diffusion, but instead of just a channel, we actually have the protein has to carry the ion across. So it doesn't just open and close like a gate, but it actually it brings something from one side to the other. And so it's usually small polar molecules that are assisted across the membrane by a carrier protein. So the protein, remember, is very, its action depends on its shape. So the binding of the substance on one side of the membrane causes a change in the shape of that carrier protein. And that change in shape is going to like pull whatever substance it was from one side of the protein to the next, and then it's going to release the substance on the other side of the membrane. And it only moves substances down their gradient. And so this is fairly similar to that leaf channel. It's just, instead of just letting things go through, it actually helps move them through. A uniporter, so like porter, I think like transporter, like we're moving things. Uni means one thing. It's a carrier transporting only one substance. So uni for one and porter for like we're moving things, like a porter moves things around. So we're moving one thing. The number of channels or carriers determines the max rate of substance transport. So this little sentence right here, I like to think of as, let's say we have a concert starting, right? And everyone really wants to go to this concert. It is really popular. And so everyone has like grouped. They're gathered outside the concert hall or wherever it's being held, and they're waiting for the doors to the concert to open. So you have, thousands of people outside, and they're all waiting for the doors of the concert to open. So if they only open one small door, it's going to take a long time for everyone to be able to get in, right? And so if we only have one channel or one carrier, then it's going to take a long time for everyone to move in. It's going to take, our substance transport is very slow if we only have one carrier. But imagine instead of opening one door, if we opened 10 doors, so then we had 10 different, points where people could get in, it's going to be a lot faster. So the more channels or carriers that we have that we can open, the faster we can transport those people inside the concert or that substance inside the cell or outside. So here's our carrier-mediated diffusion. So we have a glucose carrier protein here. So glucose is going to bind here. That's going to cause a change in shape in the carrier protein, and it's going to spit it out on the other side. And so usually these are going to be larger or polar molecules. Now, osmosis is a special case of diffusion. And we said before that diffusion is the movement of solutes, of things that are dissolved in the solvent, which is water, right? Water is the solvent of the body. And so we're moving those salts and those sugars and glucose and things like that. Whereas osmosis is kind of a special case of diffusion, where osmosis is the movement of water and not solutes. And so the osmosis is the passive movement of water through a selectively permeable membrane. So most cell membranes allow for the passage of water, but they prevent the passage of most solutes. So sometimes the solutes can't actually diffuse. So like the salts and the sugars, like if we don't have enough of these channels, glucose is not going to be able to diffuse through. But a lot of cell membranes are going to allow water to move through. And so sometimes if the solutes can't move, in order to to equalize the concentration, we can actually have the water move to equalize concentration. And so osmosis is going to happen when there's a difference in the water concentration on either side of a membrane, and the membrane is permeable to water. And so there are two ways that water molecules can cross a membrane. So even if water is polar, sometimes it can slip between the molecules of the phospholipid bilayer, but more often it's going to move through something called an aquaporin. An aquaporin is an integral protein that lets water through. So it's like a transport protein just for water. And our cells are going to have a lot of these aquaporins. And we'll talk a lot more about these in the kidney chapter too. But a lot of cells do have aquaporins, and they allow for water to move, and to move from within the cell to in the interstitial fluid, and from within the interstitial fluid to other tissues and things like that. So we have a lot of water movement in the body. Chapter 25 and 169 we'll talk more about that. And so if we look through, I think there was a picture here, we can see that this is the aquaporin with water moving through it, and some of them can squeeze through this. It's a pretty small molecule, so some of them can squeeze through here, just right through the plasma membrane. But we do have a lot of aquaporins that let more water move through more quickly. All right, so that's water. So water can move through this, its aquaporins, and it can usually move through most of the membranes. But solutes can't always move through membranes. So we have two types of solutes. So we have permeable solutes. These are the solutes that pass through the bilayer, right? Small and nonpolar substance like oxygen and carbon dioxide and urea. These are the ones in which simple diffusion happens. So these permeable solutes, that's simple diffusion. And if we look through here, we have got, I don't know if it shows, any of these moving through. I think it was just the last one, but oxygen, carbon dioxide, and urea are going to be a good example of these small and nonpolar substances. So anything that uses simple diffusion is a permeable solute, like a ghost. It goes right through the walls. Whereas non-permeable solutes are all the normal, bigger things. They are usually charged or polar or large things like ions or glucose or proteins. These are prevented from passing through the bilayer, right? These non-permeable solutes are going to be able, these are the ones that need those protein channels to help them cross the membrane. And so these permeable solutes can move to equilibrate across the membrane, but these non-permeable solutes, if they do not have a protein channel, they can't move. So sometimes, let's say we have a membrane and we have a lot of salt on one side, but not very much salt on the other, and there's no sodium or chloride channels in this membrane. The salt all has to stay on this side, and the salt is like, no, we're all cooped up, we're all stuck on this side, and we can't get to the other side, and there's not enough water. The water will actually move. The water will move towards the salt. So the water moves towards the salt to help dilute the salt out and to kind of help try to equalize those concentration gradients. And so, as we said, there are concentration gradients that exist across the plasma membrane, especially for permeable solutes, right? For non-permeable solutes, the ones that can't get through the membrane unless they're allowed. Because these non-permeable solutes are prevented from moving across the bilayer. And if we have a concentration gradient of a solute, we also have a concentration gradient for water. Because if we have, for example, let's say we have two areas that are separated by a plasma membrane. And on one side we have 30% salt, and the other side we have 70% salt. So if we look at this in percentage, if we have 30% salt, how many percentage of water do we have? Well, that means we have 70% water. If we have 70% salt, what is our remaining percentage of water? Only 30%. So because we have this concentration gradient for solutes, we also have a concentration gradient for water. So if we have a lot of solute, then we don't have very much water. And if we have, only a little bit of solute, we have a lot more water. So water can actually move down its own concentration gradient. So greater concentrations of solutes contain a lower concentration of water and vice versa. So water is going to move down its own concentration gradient. And the movement of water down its own concentration gradient is just termed osmosis. It's like a special term for it. And then water will move down its own concentration gradient until equilibrium is reached. So water will move from a solution of 1% solutes and 99% water into a solution of 3% solutes and 97% water, right? So it's going from the 99% water down to the 97% water. Now the problem with this is usually whenever we name a solution, we're not going to tell you the percentage of water in the solution. We're going to tell you the percentage of solute in the solution. So you kind of have to work backward, right? Like I said, remember, I was like, so 100 minus 1 is 99, so we'd have 99% water. And 100 minus 3 is 97, so we'd have 97% water. And so, you can do that to really understand how water is moving down its own concentration gradient. Or you can remember that water always moves towards the area with more solutes, right? Because the more solute means there's a lower water concentration. So water moves towards the salt. If there's more solutes or salt, water is going to move towards it in order to help dilute that area out. So this is going to cause something that we call osmotic pressure. And so the pressure exerted by the movement of water across a semi-permeable membrane can actually be enough to sort of counteract gravity. And the steeper the concentration gradient, the more water is moved by osmosis and the greater the osmotic pressure. And then hydrostatic pressure, but hydro is water, is pressure exerted by a fluid on the inside wall of its container. And this makes more sense in just a second. All right, so let's look at this example here. So this is a semi-permeable membrane in the middle here of this little U pipe. And on one side, side A, we have a higher concentration of solutes and less concentration of water. And on side B, we have a lower solute concentration and a higher water concentration, right? So the solutes are these black things or blue things, and the water are these, red and blue things here. So what's going to happen is the water is going to move towards the area of more salts to try to dilute it out. So water is going to go in this direction. So, if we have a little time that's going to pass, it'll start looking like this. The water has moved across the semi-permeable membrane towards side A. And now we have an equal concentration of water and solutes on either side. But because of this, like this osmotic pressure has caused, it looks really weird, right? Like this is going against gravity. Why would we have, more water in one side and less water in the other? How does that happen? It's because the pressure of water moving across the semi-permeable membrane is called the osmotic pressure. And this can actually be pretty significant. If this was a closed system here, if we had water moving into like a water balloon, it could actually put pressure on that water balloon and start stretching it or inflating it. And this comes into play when we talk about a cell, right? If we have water moving into a closed cell, it's going to stretch that cell out. If we have water moving out of a closed cell, it's going to shrivel it up. All right, so osmosis and tonicity. So this is going to be talked about a lot when we talk about red blood cells. And if you guys did the virtual lab for the cell section, we looked at some red blood cells under the microscope, and this, you guys can go back and review that, I think, if you want to, but this is going to be what we're talking about here, how putting a red blood cell in different solutions with different amounts of solutes is going to cause it to change in shape. So the cell loses or gains water with osmosis, along with a change in cell volume and osmotic pressure. All right, so what does that sentence mean? That sentence basically means if we put a cell in water, a cell has a plasma membrane, right? It has a semi-permeable membrane, and it's permeable to water. It is not generally permeable to like salt. So let's say if we put it in salt water. If we put a cell in a really high concentration of salt water that has more salt on the outside than inside the cell, water is going to want to move out, right, to dilute that out. It moves towards the area of greater salt concentration. That's going to start shriveling it up. But in the opposite, if we put a cell in pure water, there's more salt inside the cell, so water is going to try to move into the cell, and it's going to swell the cell out. And remember, we said that hydrostatic pressure is the pressure that water is going to exert on the walls of its container, so that increased osmotic pressure and hydrostatic pressure might actually cause the cell to burst. So think about overfilling a water balloon. The tonicity of a solution is the ability of a solution to change the volume or pressure of a cell by osmosis. And so we have a couple of different terms that we can use to describe these solutions, the relative concentrations of solution. So isotonic means that it's the same. It's like the same as inside the cell. Hypotonic means it has a lower solution of a lower concentration of solutes, and hypertonic means it has a higher concentration of solutes. Or if we're comparing two solutions together, the one that has the lower concentration of solutes is hypotonic, the one that has the higher concentration of solutes is hypertonic. And isotonic would mean they had the same concentration of solutes. So an isotonic solution, both the cytosol inside the cell and the solution have the same relative concentration of solutes, right? So they are in equilibrium already. So normal saline has a concentration of 0.9% NaCl, which is about the same as the inside of the cell. And this is commonly used in IV solutions because we don't want to shrivel our cells of our patients or burst the blood cells of our patients, right? That wouldn't be very good. So there's no net movement of water. A hypotonic solution is going to be like pure water. A solution has a lower concentration of solutes and a higher concentration of water than cytosol. So like red blood cells in water. So the water from outside the cell is going to move down its concentration gradient, and it's going to be forced inside the cell. As we keep overfilling the cell with more and more water, it might eventually pop. And so lysis is the rupturing of a red blood cell, and hemolysis, or rupture of something. Lysis is breaking something, usually rupture of cells, but hemolysis is specifically for red blood cells, or erythrocytes. So erythrocyte is the fancy term for red blood cells. And then finally, a hypertonic solution is a solution that has more solutes relative than the inside of the cell. And so we said that red blood cells generally have about 0.9% saline inside. So if we put them in a 3% saline, or anything higher than that, then the outside of the cell has a lot more saline. And remember that water moves down its own concentration gradient, but towards higher solute concentration. So water moves towards the hypertonic solution. And so it moves from inside the cell to outside the cell. This is going to decrease the volume and pressure of the cell and cause it to shrivel up, and we call this crenation. So this is a good picture, and this was the lab that you guys did in the cell lab, where isotonic solution, there's no net movement, the cells are happy and they are not bloated or shriveled. Hypotonic solution, water moves into the cell and it's going to swell the cells up and possibly pop them. And then hypertonic solution, water leaves the cell and it's going to cause them to shrivel up and shrink. So that is the end of our passive processes. So to review, passive processes do not require energy, and their movement of a substance from an area of high concentration to an area of low concentration down its concentration gradient. So now we have active processes. Active processes are going to be organized into two things called, one is active transport and one is vesicular transport. And we're going to talk about active transport first. So this is where it has to do with channels and stuff and not vesicles. So the active transport is the movement of a solute against its concentration gradient. So in this case, we are moving something from an area of low concentration to an area of high concentration. So we call this moving it against its concentration gradient or moving it up its concentration gradient. Because remember, I think of the concentration gradient as like a hill. So if we go from the bottom up, we're moving it up its concentration gradient. So if we're going from the area of like, you know, where we have like, let's say 2 salt particles up to an area where we have, you know, 90 salt particles, we have to move up that hill. So think about if you put the bowling ball at the bottom of the hill and try to push it up, how much energy does that take? It takes a lot more energy. So active processes take a lot of energy. And the source of energy is going to determine whether it's primary active transport or secondary active transport. This active transport here is going to be something that it creates the concentration gradients. Because if we didn't have any active transport, everything would eventually equalize, right? Because everything's always going down the concentration gradient until it equalizes. So active transport is going to create those concentration gradients. So it's going to allow the cell to like take in glucose that it needs, right? Even if there's not very much outside the cell and it's stockpiling it inside the cell. And it allows the cell to get rid of all the waste products, right? Primary active transport derives its energy directly from the breakdown of ATP. So this is pretty simple. Remember, we said ATP is a high energy molecule and that third phosphate has a high energy bond. Usually what happens is phosphorylation. So that phosphate group is transferred from ATP to the transport protein and it gives it its energy. This changes the protein shape and then movement can occur across the membrane. So ion pumps are the most common type, and we're going to talk a lot about ion pumps, both the sodium-potassium pump as well as the calcium pump. And this is a calcium pump here that we're using as an example. So cellular proton pumps are going to move ions across the membrane. And so ATP is going to bind, right, and then the phosphate is going to bind to the pump, and then it's going to cause the pump to open up, and it's going to move something up its concentration gradient. And so this is going to be important. Like let's say like calcium right here, cells do not want to have calcium inside them. If we have too much calcium inside them, you know, we could cause calcification and different things like that. So cells usually pump it out. It doesn't like to be inside the cell. And so calcium pumps in the plasma membrane of red blood cells, right? They pump all that calcium out so the cells don't get rigid, right, or calcified from that accumulation of calcium. And this lets red blood cells remain flexible enough to move through those really tiny capillaries where they have to kind of curl up. And so it's really important that we can, you know, get rid of things we need to get rid of or, you know, like take in a lot of glucose or something that we need. So these pumps allow us to maintain concentration gradients. So the sodium-potassium pump is a really important pump that we'll talk about like all the time. And it's called an exchange ion pump. And an exchange ion pump means it's moving 2 different substances, one from outside the cell into the cell, the other from inside the cell outside of the cell. But both of them are moved against their gradient. And so both of them are like active transport. They're both moving up their concentration gradient. So in this case, the cell continuously exports sodium out of the cell and moves potassium into the cell. And what I like to say that let's say the cell is like your house once again, right? And as we said before, like the door is kind of like, can be a channel or a pump, whatever you want. But things that you want in your house, you want puppies. Puppies are cute. You want them in your house. So we say potassium is like a puppy. You want potassium in the house. So the house, like we're going to go out and we're going to like, the pump is going to grab all the puppies that are outside in the rain and pull them in the house. And we're just collecting all the puppies in the house. But let's say sodium is like a snake. What do we want? We don't want sodium in we don't want snakes in our house. So the pump is going to take all the snakes and pump them outside. So keep the snakes, the sodiums stay outside, and the puppies, the potassium stays inside. And so this sodium potassium pump helps. It's going to continuously, anytime it sees, snakes or sodiums inside the house or the cell, it's going to pump those sodiums out. And anytime it sees potassium or puppies outside the cell, it's going to pull them back in. All right, so that we have all the puppies inside and all the snakes outside. So it maintains these really high concentration gradients, right? Because it's going to be moving things against their concentration gradient. So this is really important for the normal function of nerve and muscle cells. And we will eventually get to that. But just remember, the sodium-potassium pump moves things against its concentration gradient. It continuously moves sodium out of the cell, so the snakes, it throws them outside, and it moves potassium into the cell, the puppies, it pulls them in. And so specifically, we're going to kick 3 sodiums outside, and we're going to pull 2 potassiums inside. So 3 snakes are out, 2 puppies come in. And it needs ATP in order to do this, right? So this takes a lot of work, right, to pull the puppies in and get the snakes out. So we need energy to power that. So the other type, if they're not powered directly by ATP, it's powered by something else. We call this secondary active transport. And secondary active transport also moves a substance against its concentration gradient. But remember that we said that the movement of energy, like kinetic energy, we can use this kinetic energy to power something else. So the energy from the movement of one substance down its concentration gradient, so we have this kinetic energy from one substance moving down its concentration gradient, can power something else. And so, right, so remember when we talked about the electron transport chain, we said the movement of those electrons, right, we're going to power, that was powering the hydrogen pumps, and the movement of hydrogen down its concentration gradient was going to be powering the ATP synthetase to make ATP. So that's kind of a good example of that. So as something moves down its concentration gradient, that kinetic energy from that is going to be, the energy there is going to power the secondary active transport. So it doesn't directly use ATP. It actually kind of uses a, different type of energy. It's going to harness the energy from the movement of one substance down its concentration gradient to move another substance up its concentration gradient. And so this has to be coupled. So the one moving something against its concentration gradient has to be coupled with moving something else down its concentration gradient. So sodium moves down its concentration gradient. And that's dependent on the sodium-potassium pumps for energy. So sodium-potassium pumps, we pumped all the sodium out so that we have a high concentration gradient. And that concentration gradient will then allow, when we open a passive channel, it's going to allow sodium to rush back in. And that rushing motion, we can harness that energy of the sodium rushing back in to power something else. And there are 2 different types, symport and antiport. So symport is if we have 2 substances moved in the same direction, right? Sym is like same, symporters, they move in the same direction. This process is called symport secondary active transport. So they're moving, either both of them are moving from inside the cell to outside the cell, or both of them are moving from outside the cell to inside the cell. But remember, we still have one of them is moving up its concentration gradient and the other one is moving down its concentration gradient. So they're moving in the same physical direction, but still one is moving up its concentration gradient and one is moving down its concentration gradient. Whereas antiport, the 2 substances are moving in opposite directions. And that would be that one is moving from inside of the cell to outside of the cell and the other is moving from outside to inside. So they're going in opposite physical directions, as well as one is moving up its concentration gradient and the other is moving down its concentration gradient. And so if we can look at this one, this is the symport. And so we have got glucose that's transported along with sodium. So we say sodium, the snakes that are always pumped outside. So we always have this really high concentration of sodium outside the cell. So sodium always wants to rush in the cell whenever it can. And so the symport one is kind of easier to understand because we can say, well, the sodium is going to be rushing down its concentration gradient, and it actually it's going to grab onto this glucose and help it come in, right? So the glucose is actually moving against its concentration gradient, but that sodium kind of helps it come in. It brings it in. So we're using the power of that sodium rushing down its concentration gradient to power this glucose, this sodium glucose symporter. And we'll talk about this again in the kidney. Another one of this is an antiporter. And so an antiporter here, we have got, once again, sodium diffusing down its concentration gradient. And as it does this, the energy that's captured from this is going to power this antiporter, and it's going to be pumping these hydrogen ions out of the cell, up its concentration gradient. So they're going in opposite physical directions, as well as one is passive transport and the other is active transport. Whereas in the Symporter, they're going in the same physical direction, but still one is passive transport, sodium is passive transport, and the glucose is active transport. All right, so that was active transport. The one analogy that I like to use is, remember that pumps are active transport. And so if you had to like pump out, let's say the basement was flooded and you had to pump it out. If you bought a pump, would it need power? Would it need energy? Yeah, it would, right? It usually has a plug and you plug it in. So pumps need energy. So pumps they need energy in the form of, ATP or something else, right? If it's secondary active transport. So pumps need energy. So remember that pumps need energy in the form of usually ATP, because you have to plug them in. And with active transport, everything is going to be moving up its concentration gradient. All right, so the last active transport, active process we'll talk about is going to be vesicular transport or bulk transport. This is when we're moving a bunch of stuff. Usually it's big proteins. And this involves a lot of energy to make a vesicle and to transport that vesicle around. So the vesicle is like going to bleb off the Golgi apparatus, and then it's going to move to the plasma membrane and merge with the plasma membrane, and then undergo exocytosis to spit things out. So this membrane-bound sac that's filled with materials, it takes a lot of energy for the cell to move it around. And so a lot of times we'll use, ATP and things are going to be used as energy to move these things around. So we can either say the two main processes are exocytosis, which is going to be transporting things out of the cell, and endocytosis, which are pulling things into the cell. Exocytosis secretes large substances from the cell. We will talk about this a lot when we talk about neurotransmitter release from the nerve, right? The nerve is going to release that neurotransmitter from the nerve. These are big macromolecules that are too large to be moved across the membrane with like just a little protein, right? Because they're big proteins themselves. So we pack these within these transport vesicles. The vesicle fuses with the plasma membrane. This requires ATP, right? So it requires energy. And the contents are released to the outside of the cell following fusion. And this, as I said, the good example is the release of neurotransmitters from nerve cells. This one is not as much about concentration gradients as moving big things and having those vesicles. So exocytosis here, we have this vesicle, right? Let's say this was made in the rough endoplasmic reticulum, right? It's a protein. Maybe it went to the Golgi apparatus, then it was modified. Now the Golgi is shipping it out. So it's going to go up here. It's going to bind to the plasma membrane. It's going to start fusing to the plasma membrane, and then eventually it's going to release all of its stuff outside. That's exocytosis. Now the opposite of this is endocytosis, where the cell is taking in things, large substances from the outside. And so the steps are similar, but in reverse. But there's a couple of different types, and it can be used for a couple of different things. So we can take materials for digestion. So some, especially immune cells or phagocytes, and they're going to go engulf other like bacteria and things and eat them. It can regulate the membrane protein composition to alter cellular processes, and there's a couple of other things. The three different types are going to be phagocytosis, pinocytosis, and receptor-mediated endocytosis. So phagocytosis, I usually call it cellular eating. This is when a cell sticks out these big pseudopods or arms, and it looks like it's forming a giant like mouth or claw, and it's engulfing a bacteria. And so these large extension, these pseudopods, are going to surround a particle, something big, and encloses it in a membranous sac. And that membrane sac is then going to go inside the cell and will fuse with a lysosome in order to digest it. And so there's only a couple of different cell types that are phagocytic, especially a lot of them are immune cells. All right, so here's our pseudopods. It's eating this big particle. And then it's going to have this vesicle, which will eventually fuse with a lysosome so it can digest it. Pinocytosis is also called cellular drinking, and this is when the cell has lots of little, it just like internalizes lots of little pieces of extracellular fluid that are filled with solutes, so those dissolved solutes. Multiple small vesicles are formed, and most cells can do this, right? So it's going to make these little tiny vesicles, and it brings in these little areas here, and it's going to be able to bring in all those different solutes and things from the outside. But the last thing is called receptor-mediated endocytosis, and this is important if the cell wants to take in specific, like a certain thing, and especially if that certain thing is not very common, right? Because let's just say that you wanted to, you know, catch a certain fish from the ocean or something. You're not just going to take giant bottles of ocean water and like buckets and buckets of ocean water and just hope that you get that fish, because you're probably not going to. You're going to like fish for, like set bait and wait for the fish to get there and then pull it up. And so what the cell does is it puts out those receptors on the outside. And so it waits until those receptors have that specific thing it wants bound to it. And it waits, it might have multiple receptors. And when all of those are filled, let's say you have multiple fishing rods and, you know, with bait on them, and then you have all the fish on them. And then once we have all those fish, it's going to start reeling them in at once. And so this is going to enable the cell to obtain a lot of those things at once without just like indiscriminately taking lots of stuff into the cell. So a good example of this is the transport of cholesterol from blood to a cell. So cholesterol is bound to low density lipoproteins, LDLs. These are going to move in the blood, and then they're going to bind to LDL receptors on the plasma membrane. So once those LDL receptors are all full, it's going to then internalize that, that area. It's called it's coated by the protein clathrin. So in this clathrin-coated pit. So we've got these little receptors, and this does not happen. It waits. It doesn't happen until we have enough of the cholesterol, and then we've got all of that cholesterol inside, or whatever specific one that it's going to be engulfing. All right, so this is our last one. There is a good crash course video that you can watch. It's an Edpuzzle that goes through all these different types of membrane transport. Membrane transport is really important because it's important for nerve and muscle cells, and we're going to talk about it a lot. So it'll be good to understand the different types of membrane transport. Let me know if you have any questions.